Hello and welcome to today's very first review interview. It's my absolute pleasure to introduce the joint lead authors of this publication, André Görgens from the Karolinska Institute in Sweden and Bas van Balkom from the University Medical Center Utrecht in the Netherlands. We start the interview by finding out a little bit about the author's journey through science so far, what led them to this publication, a bit about the publication itself, and then what it means for the field. So we really hope you enjoy this interview, and please remember to like or comment at the end to let us know how we did.
Yeah, so I entered the EV field quite some years ago when I was working together with Willem Stoorvogel, one of the, I think, pioneers in the EV field. And then EVs to me were the unique opportunity to really have an isolated organelle and do proteomic studies on really an isolated organelle. Really, yeah, unique in nature, I think. I changed labs. Started in the regenerative medicine field, and then, of course, EVs were appearing as a very promising therapeutic agent, and pursued my career really on that, focusing on kidney regeneration, endothelial regeneration, and yeah, finally focusing on the MSCs, which I think are the most promising entities in regenerative therapeutic applications of EVs.
Started during my PhD when I did it in Bernd Giebel's lab in Essen in Germany. He was also a co-author on this paper. And we studied hematopoietic stem cells and how they divide and give rise to all the different blood cells. And we basically did cell biology on those cells and came across some proteins that were distributed asymmetrically and appeared to correlate with cell fate determination of these cells. And these turned out to be tetraspanins to some part. And then we cloned them and saw actually that if you overexpress fusion proteins, that they are like small vesicles also released by the cells. And then that was kind of 2010 or something. And at this point, we didn't have many options to analyze EVs. So I did a lot of flow cytometry before, so I kind of tried to use the instruments I had as well to study EVs or to analyze and quantify EVs, and succeeded with some instruments, especially like this imaging flow cytometry approach. Later on, I then decided, because I became so fascinated by this topic and I really wanted to understand those EVs more, how they how they facilitate communication between cells. So I went to Samir Lanalus's lab in Stockholm to learn more about EVs and EV engineering, and since then used this multiplex bead-based approach and many other engineering approaches for therapy, and also looking into biomarker discovery.
Looking at these MSCs, so the mesenchymal stem cells, mesenchymal stromal cells, they can be derived from many different sources, many different culture methods, also many different isolation methods for the vesicle. So it seemed like we were all doing the same, but still we were all doing something different. And that for me was a reason to look into these vesicles. Do they have something in common, despite the heterogeneity in MSC source, culturing, EV isolation procedures, etc. And then there was a very stimulating meeting that we had in Singapore, really focusing on MSCs. And from that, we decided to do a proteomics study without touching any pipette. We looked at papers that published proteomics data, and then we saw that despite all the heterogeneity, there was a proteomic signature of these vesicles, and that was really a hint towards, hey, there is really something that we are doing the same. This was picked up by Stefan Wild from Miltony, asking us for input on how can we develop a kit or a tool to robustly assess the quality of EV preparations from MSCs. And their goal to build this robust platform aligned with our wishes to do this inter-laboratory assessment of the kit, taking into account the heterogeneity of these MSCs and the vesicle preparations. And that's when we gathered a nice group across the world to start this effort. And I think, André, you took the lead in this.
I think we both took the lead, kind of. I mean, Bas is definitely more the expert on the MSCs than I am. But when I heard about this, I was happy to join in, and it's really I always like new toys and new technical methods that we can play with and make it as good as it gets. And the challenge of this was, of course, that we did not sit together and everybody agreed on a very, very specific way how to prepare the EVs, how to culture the cells, how to purify, how to dose the assay, and how to exactly run the assay, because there are different parameters that you can change. But then actually everybody did it like they thought it would be best. And I think this is also one of the strengths of the paper, that there are so many different parameters between all the labs. But at the end, it's possible with this assay to put the data together and normalize it appropriately, and then see the differences and similarities, and identify this common MSC-EV signature. So I think that was, it's maybe not like the most fanciest science, but I felt it's like an important thing to do, and I was really happy to see that the results that came out were comparable, even though we didn't align everything like you would do for a big ring trial. And I think that was a good first step towards defining some MSC quality control criteria, or identity criteria.
This interlaboratory effort is something that it's so important, and it's also quite scary, because everybody has his own way of working in the lab, and has his own procedures. They often, I'm for myself, I'm very conservative. It's something that when you write a systematic review, I think reading systematic reviews, one of the recommendations that you will always see is that there has to be an inter-laboratory effort, because that's hardly ever done. And that's something that's, I think the most important thing we did in this paper.
I think crucial also has been that we have a narrow question. I've participated in other interlaboratory studies before, and I just feel, of course, it's sometimes more useful if they get bigger and look at every aspect, but it also slows down things dramatically, and it ultimately sometimes doesn't get published or isn't shared with the community. And I mean, in terms of what we found out in the paper, without going into the absolute details, is that we we qualified a method that can be used to robustly assess a certain set of surface markers on MSC-EVs. And there are common EV markers and somewhat specific EV markers. What we definitely show is that the combination of different ones, so the signature overall, is quite well describing an MSC-EV. One important aspect also was that the markers we identified, of course, are the ones that were in the panel, so there might be others that are not in that panel. And also it has a certain bias towards higher abundant markers, because if a marker would be pretty low abundant, then it would be harder to pick it up robustly in all samples, even if you run the assay a bit different. So the MSC-EV marker signature is definitely consisting of like the more robust markers, because they are more abundant on those MSC-EVs.
I think it's very stimulating that everybody who is still using their own approach can pursue on their route. It's clear that doing something different is not doing something wrong. Compared to traveling to an ISEF meeting, some people go by train, some people go by plane. You could even choose to bike. In the end, we all arrive at the ISEF meeting and have a nice time. So the way to get there, the way to prepare your EVs can be different, but then the goal and the result is the same. So we're doing things different, but still we're doing things the same.
And I mean, there are so many open questions still, right? I mean, what we looked at now is like a bulk-based method for MSC EVs. Of course, there are other EVs and there are other methods. Our lab is not an MSC only lab. We work sometimes with MSCs and their EVs, but I mean, there are so many questions. As an example, we are looking into EVs derived from a lot of different cell types and try to find how different are they actually and how similar might some be. Of course, ultimately to identify phenotypic and hopefully functional differences between subsets of EVs that we can then use as biomarkers or exploit for therapies. So I think there's a lot of heterogeneity in the field overall that we don't see yet, just because we don't have the tools or the markers, or took the wrong way to go to the aim, or took a longer way, like we took the bike for some instances. And I guess in brief, biomarker discovery, because it will just come first in terms of diagnostic relevant markers that will be very specific for different diseases and hopefully pick them up early in many cases. I'm not unexcited about therapeutic approaches, but I think this will take a little bit longer to be really widespread used.
Yeah, I'm super enthusiastic about the therapeutic approaches, and but also, yeah, realistically, it's going to be very expensive, I guess, to make these EVs, especially from cultured cells, as like a standard or maybe off-the-shelf therapeutic product. There are many other approaches like the platelet derived vesicles, also other biofluids. We see milk, we see amniotic fluid, where they derive vesicles from, and they show really in small trials positive effects. I mean, there are trials going on right now, and in Austria, they're very good at these MSC EVs. If we go to the US, then there's many fresh companies with different approaches already doing experiments in humans with different applications, and also successful. So it's going on, and before it's widespread, that will take, now let's say 10 years.